Today we're diving into a revolutionary and yes, controversial topic in genetics, using CRISPR-Cas9 to edit the human genome. This is part 1 of a 4-part series where we break down how gene editing has been done in living humans. We'll explore 2 real-world cases, one from the US and one from China, looking at how they achieved in vivo editing, what made their approaches different, and why one was hailed and the other was buried in silence and scandal. But before we talk ethics, we must talk science. Today's episode is all about understanding the mechanisms behind CRISPR-Cas9 system and the introduction of some genius innovations which have improved the system's efficiency. Think of CRISPR-Cas9 as a DNA assassin, programmable, precise, and highly accurate when wielded correctly. This system relies on a molecule called guide RNA. In nature, it's made up of 2 parts, CRRNA and tracker RNA, but scientists have fused them into one streamlined version, the single guide RNA, or sgRNA. The sgRNA has 2 key parts, a 20 nucleotide spacer that is complementary to the DNA target region, and a scaffold that locks the RNA into the Cas9 protein. Just as important is the Cas9 protein. This is a multi-domain machine, like a modular robot. The 4 most important modules include the NHEJ domain, which cuts the DNA strand that is complementary to the sgRNA, the RUF-C domain, which cuts the opposite strand, the REC lobe, which ensures the sgRNA is locked in and helps scan for matching DNA sequences, and the PAM interacting domain, which scans for tiny but essential markers called PAM, proto-spacer adjacent motifs. Without this, Cas9 won't even draw its sword. To appreciate how these modules work together, let's walk through the CRISPR-Cas9 system. Step by step, I will show how this system finds and slices its genetic targets. Cas9 will load up with its sgRNA binding partner to create a CRISPR-Cas9 complex. It begins scanning the genome for PAM sites, typically the letters NGG, N being any of the codons. Once it spots a PAM site, it will unwind the nearby DNA to perform a quick compatibility check. If the sgRNA's 20-base spacer matches the DNA, boom, lock and load. Cas9 will shift into attack mode. The H&H domain will cut the strand which is bound to the sgRNA, and the RUF-C domain will cut the other strand that is opposite the target strand. Therefore, a double-strand break is made 3 bases upstream of the PAM site. The cell then immediately panics and activates its emergency repair system, and that is how the CRISPR-Cas9 system targets and cuts our DNA. But where does the gene editing part come in, you may ask? Well, that's more to do with us than it has to do with CRISPR. Here's where the magic, or rather the chaos, happens. When Cas9 makes a cut, the cell has to fix it. The most common fix? Non-homologous end joining, or NHEJ. But NHEJ is the genomic equivalent of fixing repairs with duct tape. Imagine this DNA sequence. Cas9 will cut a few bases upstream of the PAM site, between the C and G in this case. NHEJ steps in like a desperate handyman with only 20 minutes left on the clock. Sometimes it deletes a bit of the DNA, sometimes it adds random ones, sometimes it does both, creating an indel. This might be great for disabling a bad gene, but for precise fixes, it's really not ideal. When precision is key, scientists try to steer the repair towards homology-directed repair, or HDR. HDR is a less efficient form of DNA repair and is less often used than NHEJ. In HDR, the cell uses a template to repair the cut, like copying and pasting the correct word into a sentence. And here's an amazing innovation. We can provide the template that is used in the copying, pasting, so long as the template includes homology arms which match the DNA around the cut and the codons we wish to introduce into the cut. This template can be injected alongside Cas9 and the guide RNA, so when the break happens, the cell has a clean blueprint to follow. But here's a challenge. Cells genuinely prefer the more efficient NHEJ path, so scientists have found clever ways to convince cells to go down the HDR path instead. One study showed a 19-fold boost in HDR activation just by blocking NHEJ proteins, and others suggest increasing the availability of donor templates encourages cells to pick the HDR path over NHEJ, so flooding the cells with these templates is probably a good idea. Here's where things get even cooler. Standard Cas9 makes double-strand breaks on DNA, which is risky. There is a chance of off-target effects occurring, as we only have 23 nucleotides to play with for specificity. But what if we only made single-stranded nicks instead? Enter the Cas9 Nickase system, a mutant version of the CRISPR-Cas9 which only cuts one strand of DNA. By using two nickases, each targeting opposite strands at nearby locations, we create a staggered double-stranded break, but only where both nickases bind. For example, nickases with an inactive ROV-C domain will only be able to cut the strand where the sgRNA binds to. If half the sgRNAs binds to the sense region of this piece of DNA, and the other half target the antisense strand, a double-stranded break can be achieved by using two single-stranded cuts. This results in fewer off-target effects and much safer system for editing complex genomes like ours. But how do we get this high-tech toolkit into human cells? One strategy: hijack nature's own delivery trucks, attenuated adenoviruses. These engineered viruses can be used to deliver the sgRNAs, the Cas9 proteins, and even donor templates. In this episode, we broke down how CRISPR-Cas9 works. The power of this tool is immense, and the revolution has already begun. There have been two landmark attempts at editing the human genome: a US study from 2025 and a Chinese study back in 2018. The science was similar, but the global reaction could not have been more different. Like, subscribe, and share if you want to hear more about CRISPR-Cas9. I'll be working on a follow-up part for this series soon, and see you next time for more science.